What if I told you that the heavy, aching soreness you feel 2 days after a grueling workout has absolutely nothing to do with lactic acid?
Nothing at all, actually.
Right. What if instead that lingering pain is the physical sensation of millions of microscopic engines inside your legs having literally torn themselves apart, and your body is just urgently trying to rebuild the rubble?
It is a pretty wild biological reality.
It really is. Welcome to today's deep dive. Today, we are looking at something far more impressive than, any skyscraper or suspension bridge. We are talking about the incredible, highly organized architecture of your own skeletal muscle structure.
Which is something we completely take for granted.
Totally. We are going to take you on a journey from the macroscopic whole muscle you can literally feel in your arm or your leg right now, all the way down to those microscopic engines, and then back up again.
It is a remarkable biological system.
Mm.
I mean, we are looking at how this specific physical structure dictates absolutely everything in your life, from how you perform your daily, mundane movements, all the way to how your physical body ages over time.
And to do this, we are pulling from a massive stack of sources for you today. We are taking textbook anatomical science, layering in 3 centuries of historical physiology, and unpacking some incredibly recent modern discoveries that completely flipped how we understand human movement.
It's a lot of ground to cover.
It is. Okay, let's unpack this. Because to really understand how your muscle works, we have to talk about Russian dolls.
Yeah, that visual is probably the most accurate way to conceptualize the tissue. Skeletal muscle is a strict nested hierarchy.
Like a box inside a box inside a box.
Exactly. Each structure you look at is built from repeating smaller units, and they are all purpose-built to generate and transmit force in one very specific direction.
So picture opening that largest Russian doll. The outermost layer is the whole muscle itself. Let's say your bicep.
Sure.
That whole muscle is wrapped in this thick layer of connective tissue called the epimysium. But when you look inside, the muscle isn't just one solid, homogeneous block of meat. Right.
No, not at all. It is highly compartmentalized. So inside that outer wrapper, the muscle is made of bundles called fascicles.
Fascicles.
Right. You can actually see this if you look closely at shredded meat, like pulled pork. Those little stringy bundles, those are fascicles.
Oh, wow. Okay. I never thought about that.
Yeah, and each of those fascicle bundles is wrapped in its own tough connective tissue sheath, which we call the paramecium.
Got it. And then we open the doll again. Inside that fascicle bundle, we find the actual muscle fibers. And just to be clear, a muscle fiber is the individual muscle cell, right?
That is the individual cell. And yes, it has its own microscopic wrapper too, the endomysium.
So many wrappers.
So many wrappers. But they serve a massive purpose. All of these connective tissue sheaths, the epimysium on the outside, the paramecium around the bundles, and the endomysium around the cells, they don't just stop at the end of the muscle.
But where do they go?
They weave together and converge at the very ends to form your tendons. Right. That is exactly how the biological pulling force generated inside the cell physically attaches to your bones to make you move.
That makes perfect sense.
Yeah.
But the scale here is where my brain starts to struggle a bit. We think of cells as these microscopic little blobs, but the source material points out that a single muscle cell isn't always microscopic in length.
No, they can be exceptionally long.
Yeah, like in a long muscle, say the sartorius muscle running down the front of your thigh, a single muscle fiber can be over 30 centimeters long. Wait, if a single muscle cell can be 30 centimeters long, people must confuse the terminology. If the muscle fiber is the heavy-duty industrial cable, the myofibrils are the hundreds to thousands of individual copper wires packed inside it, right?
That analogy clarifies a very common misconception. People frequently collapse two entirely different levels of our Russian doll hierarchy.
Because it's confusing.
Very. The fiber is the single, elongated, multinucleated cell spanning that whole distance. The myofibril is just one of the rod-like contractile strands packed by the thousands inside that cell's membrane.
Okay, so we've opened the doll from the whole muscle to the fascicle bundle to the single long fiber down to the myofibril copper wire. Are we at the center yet?
What's fascinating here is that we still haven't reached the true star of the show.
Seriously?
Seriously. The true functional unit of skeletal muscle isn't the whole fiber, and it isn't the myofibril strand. It is a microscopic segment inside the myofibril called the sarcomere.
The sarcomere.
Yes. A single myofibril contains thousands of these sarcomeres stacked end to end, functioning exactly like train cars linked together on a track.
And these are unimaginably tiny. The text notes they are about 2 to 2.5 micrometers long at their resting length.
Which is microscopic.
Yeah, and they are separated from each other by boundaries called Z-disks.
Yeah.
So one sarcomere is the space between one Z-disk and the next.
Exactly.
And inside that space are the myofilaments, specifically the proteins actin and myosin. We've finally reached the innermost Russian doll.
We have. And because these thousands of myofibrils are running parallel to each other inside the cell, millions of these tiny sarcomeres line up in near-perfect register across the width of the cell.
Like, they are all perfectly aligned with their neighbors.
Precisely. And under a microscope, that alignment creates alternating dark bands of thick myosin filaments, which we call the A bands, and light bands of thin actin filaments, the I bands.
Okay, right.
That visual striping is why skeletal muscle is often referred to as striated muscle.
Which brings up a massive historical mystery. We have this microscopic 2-micrometer sarcomere acting as the tiny engine inside these stacked train cars. But how does it actually generate the physical force to, like, lift a couch or run a marathon?
That is the million-dollar question. And the historical journey to figure that mechanism out spans centuries.
Yeah, the text goes way back.
Right back to the 1670s, Antony van Leeuwenhoek used hand-ground, single-lens microscopes and was among the first humans to even see those striations.
Just staring through a tiny piece of glass.
Basically, yeah. But his lenses lacked the resolution to explain what actually caused them.
And prior to that, in the 17th century, early anatomists like Jan Swammerdam and Niels Stenson, often known as Steno, were doing the foundational dissection work.
Right, they proved that muscles weren't just inflatable balloons of vital spirits, which
is wild. People thought muscles just inflated with spirits to move your arm.
it was a very popular theory at the time. But Swammerdam and Steno showed they were made of bundled fibers running in consistent directions. They gave us the macroscopic fascicle concept.
But the actual inner mechanical workings remained completely invisible until the mid-20th century.
They did.
And then, in 1954, thanks to the invention of the electron microscope, the scientific community had a massive breakthrough. 2 entirely independent teams published back-to-back papers in the journal Nature.
Which is so rare.
Right. You had Andrew Huxley and Rolf Neudergerk on one team, and Hugh Huxley and Gene Hansen on the other.
And together, those papers introduced the sliding filament theory.
Okay, let's talk about that.
Well, we have to remember that before 1954, the dominant biochemical model assumed that muscle contraction happened because the protein filaments themselves physically shrank or folded up.
So older scientists thought the filaments literally shrank like a coiled spring, but it's actually more like sliding your interlocked fingers together.
That was the prevailing logic, the shrinking spring idea. The sliding filament theory was a radical departure because it proposed that the actual protein filaments do not change their length at all.
Not even a little bit.
Not even a little bit. Instead, the thin actin filaments simply slide past the thick myosin filaments. A helpful way to visualize this is to imagine a galley ship.
Okay, I'm picturing it.
The thick myosin filament is the ship, and it has hundreds of tiny little oars protruding from it. These are the myosin heads. The thin actin filament is the water.
Ah, I see.
So those myosin oars reach out, grab the actin water, pull, and then detach to reach out and grab again.
Wow. So as those millions of tiny oars pull, the actin slides inward, pulling the Z-disks closer together, which shortens the entire sarcomere train car.
Exactly. And Hugh Huxley actually spent the 1960s using X-ray diffraction to map this precise geometry. We get the A band, the I band, the H zone in the middle where there's no actin, and the M line anchoring the myosin in the center.
It sounds like an incredibly precise piece of machinery.
It really is. And the physical proof of this geometric relationship came a decade later. It remains one of the most elegant experiments in all of physiology.
This is the 1966 study, right?
Yes. In 1966, A.M. Gordon, Andrew Huxley, and F.J. Julian published a study where they managed to isolate and stretch single frog sartorius muscle fibers.
Just setting up an apparatus to stretch a single microscopic cell and measure its pulling force sounds impossible to me.
The ingenuity of the methodology is astounding. By stretching the fiber to different, highly specific micrometer lengths, they proved that muscle force isn't some magical, mysterious property of living tissue.
It's just geometry.
It is a strict geometric consequence of filament overlap. They found that force peaked perfectly when the sarcomere length was between 2.0 and 2.2 micrometers.
Because at that exact microscopic distance, the overlap between the thick myosin ores and the thin actin water is optimal.
Exactly right.
It's the perfect distance for the maximum number of ores to reach out and form what the text calls cross bridges, So what happens if you stretch the frog cell way past that 2.2 micrometer mark?
The force drops entirely to zero.
Zero.
Zero. If the sarcomere is stretched too long, the myosin ores literally cannot reach the actin. There is no physical overlap to form those cross bridges.
We're just rowing in empty air.
Pretty much. And conversely, if the sarcomere is compressed too short, the thin actin filaments from opposite ends crumple into each other, and the thick myosin filaments jam violently up against the Z-discs.
So it becomes a physical, structural gridlock.
Complete gridlock.
Okay, this microscopic engine is brilliant. But if every skeletal muscle uses this exact same sarcomere blueprint, how does the body engineer these identical units for totally different, massive physical tasks?
What do you mean?
Well, my eye muscles darting around to read a book, and my calf muscles holding my entire body weight all day, they are definitely not doing the same job, but they use the same engines.
They do. And to understand that, we have to travel back up the Russian doll hierarchy to the whole muscle level. The body alters the geometric arrangement of those fascicle bundles.
The architecture itself.
Right. The architectural layout is a mechanical engineering choice with serious physical trade-offs.
Let's look at that architecture. The sources talk about parallel or fusiform muscles.
Yes.
The sartorius in the leg is a great example again. The fascicles run roughly parallel to the long axis of the entire muscle. That design heavily favors range of motion and the speed of contraction.
Compare that to a pennant muscle, like the soleus in your lower calf. In a pennant muscle, the fascicles do not run straight end to end.
How do they run?
They're arranged at an angle to the tendon, looking very much like the slanted bristles on a feather. The human soleus has a pennation angle of around 25 to 30 degrees.
But doesn't angling the fibers mean they aren't pulling directly in the line of action? That seems incredibly inefficient if you want to pull something straight up.
It seems completely counterintuitive until you look at the spatial mathematics.
Okay, math time.
Angling the fibers means each individual fiber loses some of its direct pulling efficiency, true. But that angle allows the body to pack vastly more fibers into the exact same volume of space.
Oh, because they're stacked diagonally.
Exactly. The soleus has an incredibly high physiological cross-sectional area. It deliberately trades contraction speed and range of motion for the ability to pack in more tiny engines.
To sustain massive forces to keep you standing upright for hours.
You got it.
And it's not just the architectural angle of the bundles, is it? It's the metabolic machinery packed inside those cells. Skeletal muscle fibers come in entirely different profiles.
They do. The two primary categories are type I and type II fibers. Type I fibers are your slow-twitch, highly oxidative cells.
The endurance guys.
Right. They are highly fatigue-resistant and packed with a protein called myoglobin, which binds oxygen. Type II fibers, on the other hand, are your fast-twitch, glycolytic cells.
And they don't have as much myoglobin.
Very low levels. They rely on different metabolic pathways. They generate quick, explosive power, but they fatigue almost immediately.
Here's where it gets really interesting. The source material points out a concrete, everyday example of this that you can actually see on your dinner plate. Chicken meat.
A classic example.
So you're telling me a chicken breast is essentially built for a sprint, which is why it's white, fast-twitch, type 2 meat, and the leg is built for a marathon, which is why it's dark, slow-twitch, type I meat. We can literally see this anatomical structural difference on our dinner plates.
That is a perfect demonstration of structure following function. The pectoralis muscles of a chicken, the breast meat, require short, explosive bursts of wing flapping to escape predators. Because type II fibers lack oxygen-rich myoglobin, the raw tissue appears pale or white. But the legs are dominated by type I's slow-twitch fibers, heavily saturated with myoglobin and a dense capillary supply to continuously deliver oxygen.
Because they walk around the yard all day.
Exactly, making the meat dark. The fundamental sarcomere engine inside both tissues is identical. The body simply alters the ratio of fiber types, the mitochondrial density, and the architectural angles based entirely on the functional demand.
That is so cool. But wait, earlier we talked about the sliding filament theory and how pulling the muscle too far apart drops force to zero because the myosin ores can't reach the actin.
Right, the 2.2 micrometer limit.
If the entire mechanism relies on these floating filaments not sliding too far apart, why doesn't a muscle just snap in half when I stretch my arm out? Like, what keeps them from just floating away from each other?
That specific question drove a massive shift in modern physiology. For most of the 20th century, scientists viewed muscle structure as a passive mechanical scaffold.
Like the filament slid and everything else was just inert plastic wrap holding it together.
Exactly. But the discovery of a protein called titin in the late 1970s changed everything. Titin is the largest known protein in the human body,
The largest.
by far. It spans half the length of the entire sarcomere, anchoring the thick myosin filament directly to the Z-disc.
So it's acting like a bungee cord.
Precisely. Research in the 1990s, pioneered by scientists like Hank Granzier, proved that titin is not passive scaffolding. It acts as a massive molecular spring.
Oh, wow.
When you stretch a muscle, titin generates passive tension to prevent those filaments from tearing apart. It essentially sets the resting length of the sarcomere and may even act as a physical length sensor, communicating mechanical strain to the rest of the cell.
And that changes the whole paradigm. It means the structural framework itself is active.
Very active.
And that brings us to satellite cells. The anatomist Alexander Morrow identified these stem cells structurally back in 1961. They just sit there quietly between the muscle fiber membrane and the endomysium wrapper.
Morrow found them, but the scientific community spent decades fiercely debating what they actually did in adult humans.
Right, they weren't sure if they did anything at all.
It wasn't until the 2000s and 2010s, utilizing advanced genetic lineage tracing, that researchers definitively proved these satellite cells are the absolute center of tissue repair and adaptation.
Wait, if satellite cells are just sitting there waiting to rebuild things, does that mean our muscles are constantly under construction, not just a fixed set of cells we're born with?
If we connect this to the bigger picture, it requires us to completely redefine what skeletal muscle is.
How so?
It is a highly dynamic, sensing tissue that constantly remodels itself based on the loads you place on it. Even those connective wrappers we discussed earlier, the endomysium and paramysium, are now understood to be active, signal-transducing participants that adapt to mechanical stress.
Because this tissue is constantly adapting, understanding these structural rules totally upends how we experience exercise, aging, and even just everyday pain.
It really gives you a new lens to look through.
Let's do some myth-busting with these structural facts. Starting with my opening question about DOMS delayed onset muscle soreness. People religiously believe it's lactic acid burning in their legs 2 days after hitting the gym.
The physiological reality is much more violent.
Yeah, you mentioned that.
Lactic acid clears out of your bloodstream very shortly after exercise stops. That deep, lingering soreness is the sensation of microscopic structural disruption.
The tearing.
Right. Intense mechanical loading literally tears the Z-discs and disrupts the sarcomeres. Your body initiates a localized inflammatory response to sweep away the cellular debris, and then activates those satellite cells to rebuild the architecture stronger than it was before.
Okay, here's another one. When I lift heavy weights specifically to get bigger muscles, I'm causing all that micro-tearing so my body will manufacture brand new muscle cells to handle the weight next time, right? That's what hypertrophy is.
Not exactly.
Yeah.
You are confusing hypertrophy with hyperplasia.
Oh, what's the difference?
In humans, lifting weights does not typically generate entirely new muscle cells. Hyperplasia, which is the creation of new fibers, is documented in certain animal models, but remains highly contested and weakly supported in adult humans.
So I don't get new cells.
No. When your muscles grow larger, which is hypertrophy, your body is actually packing more and thicker myofibril strands into the existing cells you already have. The fiber itself balloons in volume to accommodate the new hardware.
Wow.
Yep.
Okay. And we also need to clear up the confusing terminology around the word striated. I read articles all the time that use striated muscle and skeletal muscle as if they are the exact same thing.
It is a frequent point of confusion. Striated simply describes the microscopic visual striping pattern of the sarcomeres.
The dark and light bands we talked about.
Exactly. Your cardiac tissue, your heart, is also heavily striated because it utilizes the exact same actin and myosin geometric engine. But cardiac muscle is structurally completely different from skeletal muscle.
Right, because cardiac cells have branching networks. They are linked by specialized junctions called intercalated discs, so they contract as a single unit. Plus, they usually only have one central nucleus, and most importantly, you don't have to consciously think about beating your heart. It's involuntary.
Exactly the distinction. So while all skeletal muscle is striated, not all striated muscle is skeletal.
So what does this all mean? You mean to tell me all those fitness programs telling you to keep your muscles guessing to avoid plateaus are structurally bogus? Sarcomeres don't have brains to get confused?
This raises an important question about how our bodies actually register adaptation. The marketing claim is entirely unsupported by structural anatomy.
It's just marketing.
Totally.
Yeah.
The sarcomere does not possess the sensory apparatus to recognize whether the tension pulling on it originates from a dumbbell, a kettlebell, or a heavy box in your garage.
It only responds to the ruthless laws of physics: mechanical tension, metabolic stress, and progressive overload.
Yes. If you apply consistent, progressively heavier tension to the muscle, the satellite cells will remodel the tissue. Now, switching exercises might keep you mentally engaged or help avoid a repetitive strain injury in your joints, but it is absolutely not biologically required to prevent your sliding filaments from sculling out.
That is so freeing to know.
And understanding that mechanical reality becomes critical rather than just optimal as we age, the literature places heavy emphasis on sarcopenia, which is the progressive age-related loss of skeletal muscle mass and strength.
Which is a huge deal.
It is a primary driver of frailty and the loss of physical independence in older populations.
And the scary part is that it's not just a general shrinking of the whole muscle, is it? It is a highly targeted structural loss. The sources note that sarcopenia involves a preferential atrophy, a shrinking and loss of those type 2 fast-twitch fibers.
Yes, the explosive engines are the first to go.
Which is terrifying.
Which is why the medical community now views rigorous resistance training as essential geriatric medicine. By deliberately applying heavy progressive tension, you force the tissue to counteract that specific structural decay.
You're forcing it to rebuild.
Exactly.
Yeah.
You preserve the sarcomere integrity of those type 2 fibers, quite literally maintaining the physical machinery required to catch yourself if you stumble.
It is genuinely incredible. We have journeyed all the way from the visible, heavy architecture of a whole muscle, down through the connective tissue fascicles into the individual foot-long fibers, past the myofibrils, and right down to the tiny 2-micrometer sarcomere engine.
It's quite a trip.
We've watched thin actin filaments sliding over thick myosin ores, pulling those Z discs together to power every single physical action you take in a day. Understanding this nested Russian doll structure empowers you. You can evaluate fitness claims with a critical eye. You understand exactly what is happening when your legs ache after a run, and you can truly appreciate the microscopic engines keeping you moving through life.
It is a remarkable system of dynamic living architecture, constantly rebuilding its own steel girders. But there is one final, rather haunting structural reality derived from our source material regarding what happens when this entire system finally powers down.
Oh, this is the part about rigor mortis, right?
Yes. Throughout your life, the release of those thick myosin ores from the thin actin filaments requires energy, specifically a chemical molecule called ATP.
So you need energy just to let go.
Yes, ATP is what allows your muscles to detach, relax, and reset after every single contraction. It is the chemical freedom to move, to dance, to breathe.
Okay.
But after death, the body's production of ATP stops entirely. And when that energy runs out, the myosin ores can no longer release their grip on the actin.
They just get stuck.
The cross bridges lock perfectly and permanently in place. The entire muscle stiffens. Your final physical state, which we call rigor mortis, isn't actually a structural breakdown.
It's just a pause.
It is quite literally a permanent pause in the sliding filament cycle. The exact same microscopic handshake that allowed you to move your entire life is what locks you perfectly still at the end of it, simply because the energy ran out.
It's an unbelievable mechanical reality. From a bustling biological city constantly under construction to a permanent locked standstill, that is going to stick with me. Thank you so much for joining us as we took this incredible journey inward. We'll catch you on the next deep dive.